Aloha, everyone. Thank you for coming. We're very excited to talk about the work that the state is doing towards coral restoration. Oops, sorry. Okay, so first I want to talk about why we think and why the state thinks Hawaii needs a coral nursery. The formation of our nursery was driven by the need to mitigate for damages to reefs among our islands. So, for example, we've had various ship groundings in the past decade. They have damaged large areas of reef. We've also had things like the molasses spill just in 2013. Again, that these unplanned events are harming some of our coral reefs. And since Hawaii's coral reefs grow so slowly, it's very difficult to have any sort of natural recovery for these large areas that have been damaged. So again, why do we care about Hawaii's coral reefs, or why are they worth so much? They've been estimated to be worth $36 billion a year. And this is not because they look pretty, but I guess that is part of it. But there are reefs here, and reefs around the world have very high biodiversity. They provide for food for the people that use their resources. Our visitors come here to come see our beautiful reefs. They also, especially in Hawaii, they provide coastal protection. So when you have the waves breaking out on the reef offshore, they're breaking out on the reef and not on shore, and those reefs help protect our shoreline from coastal erosion. There's also a lot of research going into various reef organisms. So this is a lot of text, but basically what we want to get across is that corals are fully protected in Hawaii. You cannot take corals, you cannot break them. So corals have great protections in Hawaii. However, when something happens, like say ship groundings, all of those protections, no matter how great they may be, aren't really going to help that reef recover. And so that's why we're kind of stepping into our mitigation process to give these reefs a helping hand.
Yeah. So what is a coral nursery? Not those. Kind of. So there are several coral nurseries around the world whose goal it is to grow corals faster than corals would grow themselves in the wild. This is often used with fast-growing branching species. So as you can see here, the Coral Restoration Foundation in Florida, the Coral Restoration in Florida uses these trees to grow fragments of the fast-growing branching species out in the open ocean. A lot of these efforts are focused on Florida, the Caribbean, and in the South Pacific. They're kind of like plant and baby nurseries in that humans are aiding the growth and production of a species. In-water nurseries, the advantages are they are low cost and they can produce thousands of outplants. Some of the disadvantages are their outplants are very small and they primarily focus on the fast-growing branching species. And gravity plays a part in their growth form. If they're grown out on these trees, they grow differently than they would if they were in open reef habitat. Should be the next one. Okay. They require fast-growing species that can grow 15 to 20 or 25 centimeters a year in relatively calm water with low nutrient environments. And they can produce 1,000 colonies. So Hawaii corals are unique. We tried to do the tree form with Hawaiian corals, and it yielded pretty poor results. One of the reasons is Hawaii corals grow very slowly, about 1 to 2 centimeters a year. And we lack the fast-growing branching species that is found in other parts of the world. We lack that here in the main Hawaiian Islands. And our reefs are very close to shore, so the pollution and damage is directly affected our reefs. So Hawaiian corals are more of the boulder mounding formation, and they grow about 1 to 2 centimeters a year. Although they do have a slower growth rate, they provide a very stable and firm reef base. And we found that larger coral colonies do better. And this goes into the concept of a size refuge. So size refuge is a concept that nature has made a certain size for any given multicellular organism, that instead of putting all their energy into growth, they've reached a size where now they can start focusing on sexual reproduction too. So with Hawaiian corals, that size refuge is about 20 centimeters. So for example, humans, it's about 12 years old, or for dogs, it's about 8 months. So if Hawaiian corals only grow 1 to 2 centimeters a year, it's a number of years before they can even reproduce sexually. And before then, they're focused on growth. So they might not be able to handle what nature throws at them.
So I'll start getting into our nursery. So the state of Hawaii recognized a need for a unique facility here. As Honor was explaining, we can't really do the coral tree method. It just doesn't really work here with the corals we have in our conditions. So we needed to come up with another way to grow corals quickly, specifically Hawaiian corals for Hawaii, to mitigate for damages. So this is a picture of us standing in one of our coral tanks here, and our nursery is located at Anuenue Fisheries Research Center, which is out on Sand Island, or right next to the Coast Guard base. And unlike those in-water nurseries with trees, we have found that in order to do this coral restoration in Hawaii, we needed to have a land-based nursery, which we'll get into more in a minute, and so that we can fast grow Hawaiian corals in a nice controlled 5-star hotel environment. So again, our goal for our nursery is to take small corals and grow them bigger fast, right? So what we do is we take a 10 centimeter coral and we turn that coral into a 42 centimeter coral colony in about a year. And again, in the wild, that's equivalent to about 20 to 25 years of growth that we're able to do in 1 year. So it's pretty exciting. So how we do it is, this is our basic outline of our process, which we'll go into more in depth in this talk. But first, we are collecting our corals. We collect our corals. They go through a strict quarantine process. So we don't want to be introducing or spreading around invasive species or pests or heavy metals, anything like that. Then we fragment them, we grow them out to that 42 centimeter size. We acclimate them back into conditions that they would experience in the wild or where they're going to be outplanted. And then we outplant those corals onto those damaged areas that we talked about before. And we continue to monitor our corals to see how they're doing in the future. So first step, coral collection. So I think something that's very different about the Hawaii Coral Restoration Nursery is where we get our corals from. So we don't just go out onto the reef and collect a bunch of corals. We want to have the least impact on our natural reefs, right? Because we're focused on restoring them. So we target corals that grow in harbors. And this is because corals in harbors have a lower value, essentially ecological functions and services. They don't provide the same services as they would out on a natural reef. So for example, if you have a coral that's growing on a piling, a man-made surface in a harbor, if you think about those ecosystem services and functions we talked about before, it is not contributing to building reef structure. It's probably not really contributing much to a fish habitat. It's definitely not contributing to coastal protection, which is a big thing here in Hawaii. And so it's essentially not worth the same as a coral out on the reef. So that's why we use harbor corals kind of as our source corals for our nursery. Now in the exception would just be if we cannot find a species in a harbor. Not all coral species grow in harbors. They can be a little yucky. So some species prefer the nice reef outside. And so if we do need a species from a natural reef, what we'll do is we'll mitigate for our own collection. So what we'll do is if we need to take a coral from a natural reef, we will take double the amount that we normally would have. So we normally take 10 centimeters. So we'll take 20 centimeters from that natural reef, which at first sounds a little weird, right? Don't worry, I'm getting there. Mm-hmm. And what we will do is we'll take that coral, we'll separate it into two 10-centimeter pieces, and each of those 10 centimeter pieces, we will grow into 42-centimeter colonies. Then one of those colonies we will place back on the original collection site, and one colony we will use for our mitigation site. So it's kind of like we're essentially borrowing some coral for a year, and when we outplant that module back at our collection site, we're actually giving additional growth to that site because we started off with 20 and now we're at 42. So that's how we kind of even out collecting from natural reefs. And throughout this whole process, we are very meticulous about documenting all of the corals we've collected. We take photographs of before we collect the coral. We have a scale bar so we know how big it is. We take a picture afterwards. And as we'll get into later, we track all of our corals throughout their journey at the nursery.
Thanks. Sorry. Once the corals are collected, we bring them back to our facility where we look for alien invasive species that can be harmful to the coral or infest our tanks, pests, and we cut away excess rock. There's one more. Okay. So in our quarantine systems, every coral that is collected, we keep in our quarantine systems for a month. If we find any disease or any pests on that coral on day 29, we restart our quarantine process and have strict monitoring on those coral colonies. Our incoming water for our quarantine systems is filtered from the harbor, and our effluent for those systems are treated with a UV sterilizer. So the water that we bring in or the water that we put back in the harbor is much cleaner than the water that we have brought in from the harbor. And each coral colony that we collect receives a unique physical tag, and it follows the coral through the nursery. And that physical tag gets entered into our database, and our database keeps track of every coral that we've ever collected, when and where it was collected, how it traveled through the nursery, if it was fragmented and grown out onto a module, and when and where that module was outplanted, and our current monitoring updates.
So again, back to that 10 centimeter to 42 centimeter. So this is an example of, sorry, I'll use the mouse. This is an example of one of our 10 centimeter colonies. So this is our original colony. You can see it has the tag on it. There's not, it's been cleaned, and we micro-frag that. So we cut it up into a bunch of tiny pieces. So since coral is a colonial organism and all the tiny polyps, we have just tiny pieces of coral that are essentially another piece of tissue of that coral. And we take all of those pieces from that original 10 centimeter and we glue it onto this concrete module, which we'll talk more about in a second. And then we're able to grow that out at our facility, and all those pieces grow out. They meet up with their neighbor and they fuse together. And so that is how we're able to get this 42 centimeter colony from a 10 centimeter colony in about a year. And so the way we do that is with a saltwater bandsaw. So it's a little bit similar. You think of kind of a tile saw, but maybe not as scary. And it runs salt water through it. So we're able to kind of wash the corals as we cut them, and we're able to get very precise cuts from these corals. So as you could see in the previous picture, they're kind of little rectangles or little squares. So they have 4 sides in which they can grow off of. We then attach those little pieces to our concrete module with human surgical grade super glue gel, which sounds a little weird, but you know, if it's good enough for humans, we're hoping that it's good enough for coral. And so we're able to attach that onto the module, and we use that saw again to cut the bottom of the coral. So we want to cut off most of that underlying skeleton. We only want to keep minimal skeleton. So these corals have been growing in harbors, right? Harbors are not, the nicest, cleanest water. So we want to make sure that in case there's any chemicals or heavy metals or anything inside of that extra skeleton, we just want to cut that away. We just want to get rid of it, and we just want to have the most recent growth and that coral tissue on top. So you can see some of the pictures here of cutting the coral, and then we kind of have an assembly line set up. So someone else will be gluing the coral onto a module, and that goes into our tanks. So again, for micro-fragmentation, we have all these tiny pieces. It gives that higher growth rate, since it can grow on all these sides instead of just one big colony with just the edges. Again, they're all from that same source mother colony. So at the genetically identical frags, as they grow, they'll meet up and they'll fuse together so that they don't have to grow a large surface area. They only have to grow a little bit until they touch their neighbor. And again, yes.
So when you're gluing them,
Mm-hmm.
are you gluing them wet onto?
Yes. Mm-hmm.
And the super glue will just cure when it's wet?
Yes, so the super glue gel, when you put it underwater, it's not regular super glue, it's a super glue gel, it's cyanoacrylate. And when you dunk it underneath the water, it kind of forms this, seal you could call it. So we're not having anything leach out into the water. Mm-hmm. And it actually cures very quickly, usually within a minute. Mm-hmm. So yes, we're able to use it underwater to glue the fragments onto the concrete.
How long does it take to dry? Or does it take
Yeah, it's again about usually like a minute. It's very quick. Yeah.
Is it like regular super glue? Have you guys glued your fingers to the?
It glues, yeah, it'll do
yeah. We've definitely gotten glue all over our hands. Yes. Yes. It's not quite as bad as regular super glue because it's that gel, so you can kind of just pick it off. Yeah. Yes.
You have a lot of pieces there, so after the quarantine period, you don't mind mixing corals anymore? They're all put together in some larger space?
Yeah, so this is after they've gone through quarantine. We only move corals inside after they've passed our 30-day quarantine period. So that means that we usually check them daily for any pests or anything that we don't want on the coral. They're disease-free because we don't, again, like you said, we don't want to mix corals where one has an issue and it might spread to other colonies.
And on one given module, it's from the single coral colony, so it's the same genotype.
But there's no difference in requirements of salinity or temperature from the different kinds of corals that you might be growing?
There's not usually temperature and salinity, no, because they're all usually from the same area, and temperature and salinity doesn't vary that much. But we do use species-specific techniques for cutting. So some corals we can cut them really small. Other corals we need to keep them at a larger size so that they continue to do well. So yes, we do some species-specific care in terms of cutting as well as feeding and stuff like that. Yeah. Mm-hmm. Any other questions about this? Good. Okay. So one last thing about microfragmentation is, so we have all these tiny pieces, right? And we do have them in an aquarium, a tank, so it's a controlled environment. But just in case something happens to one of those pieces, say one dies or is damaged somehow, or just something happens, we still keep a little bit of that same source material from that mother colony, so that we're able to take a piece from that and replace any broken or damaged ones on our main module. So we're still able to keep that same rate of growth. Usually not that often. Most of the times, before they start growing on, sometimes we can accidentally knock them off when we're cleaning them, but we just glue them back on. Yeah.
Or sometimes urchins, we have urchins that work in our tanks to, eat algae, and they bump sometimes. But usually we catch it, we do checks multiple times a day. So usually we catch it. And once they've grown onto the concrete module, there's not a lot of bumping going on.
Mm-hmm.
So our coral modules are cast from Portland type 2 cement. Again, our goal is that 42 centimeter pyramid shape. So the 42 centimeters goes back to that idea of a size refuge. At 42 centimeters, we're basically ensuring that the coral will do well once we outplant, because they're past that size where they can start focus on reproducing instead of speedy growth. And we've chosen that pyramid shape after trial and error. The pyramid shape allows for coral to grow fast on the flat surfaces. And once outplanted, that shape is best against wave action and allows minimal sediment accumulation. So once we cast the concrete module, we cure it in fresh water for a minimum of 2 weeks, allowing any harsh chemicals to leach out and for the pH to neutralize. These are grow-out systems. You can see our concrete modules with little micro-fragmented pieces of coral growing on them. We have special LED lights that allow that have the optimal spectrum for growing coral. And we have pumps to replicate wave or surge action. Our water is filtered, and we run water quality tests to ensure that our water chemistry is in desired parameters. And to keep these systems stable, our staff are professional aquarists trained in keeping coral alive.
So another thing that we do, so again, kind of our corals are in our large aquarium systems. But we also want to make sure that they are growing as fast as possible. So they kind of already are in this, nice clean water environment. They have great lighting. They have simulated wave action. They have aquarists taking care of them all day long. And but we also want to give them any opportunity to grow faster. So what we do is we culture our own live food at our facility to feed to our corals. We also make a custom mix of foods. So we did some experiments to see what foods would be best for our corals in general for best growth. And we also just did some recent experiments with what mixtures and what types of food would be best for certain specific species. We also add calcium and other minerals to the water. So this is some of our dosing setups. So we have a machine that will automatically add calcium and other minerals to, like magnesium, to our water, so that our corals can use that to build their skeletons so they can grow as fast as possible.
So once they are 100% covered or over our concrete module, which takes about 8 to 12 months, we move them into an acclimating tank, which Christina mentioned earlier. Whoops, sorry. Okay. These acclimation tanks are outside, and they are kind of they're adjusting our corals to more of the environment that they'll be outplanted on. So we mentioned the 5-star hotel where they were getting fed by mom every day, and this is kind of like the 18, you're on your own. You got to feed yourself. And so we adjust them to light levels and surge action in our acclimating tanks. And we keep them in these acclimating tanks for at least a month before they're outplanted.
Oh, yes.
What's the survival rate of the corals? Is it pretty high going into the...
I'm glad you asked, because we're about to get into that. Yes.
Is that all different types of corals in this picture here, or is just one?
That's all different species, yeah.
Is there a problem with them, going through their reproduction and getting their coral on the other corals, and now it's combined?
So as far as just growth, we keep them separate. So we make sure they're not touching, so they don't compete with each other that way. And then in terms of like spawning, so for example, we actually have the Montipora capitata spawning coming up next week. And so we will be isolating those colonies just to see if they will spawn. And also if they do, we want to try and collect some of the spawn. Mm-hmm. But yes, so corals like that, we are able to for capitata, we're able to predict, the nights that they will spawn. But even in general, even if, say, some other species spawned in the tank, we constantly have new fresh water running into the tanks. So that would get flushed out, and it shouldn't have a negative effect on the other corals. Okay, so after they're 100% covered, they've been acclimated, they've been kicked out of the hotel, and now they're ready to go back out into the wild. What we do is, again, we want to outplant these corals onto damaged reefs. So this is kind of our method here. First, we want to go, and usually we're on scuba. We go and look at the damaged area, the site that we want to outplant our corals. We photograph potential areas that we want to put our modules, and then we go out again with our dive team, and we will outplant our modules. So they're concrete, so they're already pretty heavy, but we also use marine safe epoxy. So we will put that to secure on the four corners of the bottom of the module so that it doesn't get tipped over by wave action. Then, of course, along with just how we took pictures of the coral throughout its process, throughout its time at the nursery, we will continue to document the health of that coral once it's been outplanted onto the reef, so we can keep track of our survival and just the general health of our modules. So this was our pilot site over by Sand Island Beach Park. And you can see here there's one, sorry, use the mouse. Here there's one module here, there's another one here, here, all the way in the back. So we outplanted a total of 9 modules at this site. And we have had 100% survival. All those modules are doing great. And I think the key to that is making sure that we have had 100% coral cover on those modules. We don't want gaps in there, just in case, some algae can start growing in there or anything else. So we want to make sure they're 100% covered. And you can see here at the bottom of this one that the coral is starting to grow down onto the reef substrate. And this is one of the corals. So again, this is a little example of our monitoring. So this is after 1 month of outplanting. You can see this was in 2017. This is after 9 months. You can see it's starting to take on a little bit more of that natural shape. It doesn't look it still has that pyramid shape, but it's starting to grow down onto the substrate and become a little bit more of that boulder mounding shape. And this was actually just in a few weeks ago when we went out to monitor our corals at Sand Island. And you can see that it still has that vague pyramid shape, but it's really starting to take on that, that natural boulder shape. And you can see it's still continuing to grow down onto the substrate. So our modules at Sand Island have been doing great. Another recent outplant that we did was we partnered with the Coral Restoration Foundation in Florida. They have a day called Coralpalooza, and that's what it's called. And and it's a day for coral nurseries around the world to outplant corals. So we participated in that. Honor's going to be talking a little bit about some cool things we did with that in a minute. But we also outplanted some modules right over by the airport reef runway, where the Port Royal ran aground. So, sorry.
So part of that event, Coralpalooza, we did that one outplanting off the reef runway, and another one that we did was in Kaneohe Bay. So in 2015, in anticipation of the coral bleaching event, we collected 3 pieces of Hawai'i DAR. This is an endemic species of coral that we've only found in Kaneohe Bay. So those 3 pieces we collected, we brought back to our facility, and we started growing out. We went back after the bleaching event, and we didn't find any survival from the parent colonies that we collected from. So we assumed that they were dead. The 3 pieces that we collected were growing so well at our facility that we fragmented and we grew out onto 3 10-centimeter modules, the micro-fragmented pieces. And then a couple weeks ago, with partnership with the Coral Restoration Foundation's event Coralpalooza, we outplanted 2 of the 3 10-centimeter pyramids back into Kaneohe Bay. And from our knowledge, this is the first reintroduction to Kaneohe Bay of an extirpated coral species.
So kind of going off that, Hawai'i Sea Grant, why we're recollecting it in the first place. So one of our additional projects that we have besides our fast coral growth is having a arc of rare Hawaiian coral species. So we have a total of 40 species at the nursery, and we fast grow about 12 of them. And the 28 are rare and uncommon corals that we hold at our facility. So it's kind of like a seed bank, but for coral. So just in case something happens to these wild populations of these rare species, just like the Duardeni in Kaneohe Bay, we have at least one representative species, one representative colony at our facility. And so that really came in handy for when we couldn't find any Hawai'i Duardeni in Kaneohe Bay, and we were able to reintroduce it because we had originally collected those 3 pieces. Yes?
Are all coral species capable of being fast grown, or are some more reactive to those techniques than others?
Yeah, so some species do grow faster than others. In general, Hawaiian corals still grow really slow. They still generally have that 1 to 2 centimeter a year growth rate compared to other places in the world where it can be 15 to 25 centimeters a year. But yes, some species do grow a little bit faster than others. So we do take that into account in our fast growth protocol.
I'll talk later about another species of Hawaiian corals that we're trying to fast grow that we haven't yet.
And so one last thing about our rare Hawaiian corals is we also partner with Maui Ocean Center. And so we have a rare Hawaiian coral arc and they have a rare Hawaiian coral arc. So when either of us gets a piece of rare coral, what we do is we fragment that coral and say if we collected it, we will give them 1 piece, we will keep 1 piece. And if it's one we haven't gotten before, we will have another fragment that we sacrifice for identification purposes. And so we each have a piece of this rare coral. So if something happens to our facility or something happens to their facility, we essentially have a backup of this rare coral. Also, our facility may be a little bit different from theirs. And so we're able to compare notes and see, maybe this type of flow or lighting or care for this coral does really well for this certain species. And, we can compare husbandry techniques for different species of coral. So it's a really cool partnership that we have. So another example would be in last year, as you all know, the Kapoa tide pools on the Big Island got run over by lava. And unfortunately, the beautiful corals there are just, you can't compete with lava. So the beautiful corals there were destroyed. But before this happened, we actually had sent a technician to the Big Island to go get samples of these corals from these tide pools. So as far as we know, we are the only place that has pieces of these corals. Anywhere else, they are completely destroyed in the wild. So we only have them at our facility. And so one of our next upcoming projects, I'll let Aaron talk about.
So as we talked about earlier, for Hawaiian corals, bigger is better. They're able to produce more or, to focus their efforts more on sexual reproduction than trying to grow super fast and spend all their energy. So bigger is better for Hawaiian corals. So one of the projects that we have right now is we want to grow a 1 meter coral. So we have corals growing, as you can see here on our prototype, which is comprised of 4 parts. Each one of the 4 parts represents a side of a 1 meter sized pyramid. There's our coral growing on our perspective module. And when we outplant, we'll place all those 4 parts together. And to fuse along this line, it will only take the coral about a week. So in a year-ish and a week, we can form a 1 meter long coral in about a year, which represents about 100 to 150 years of coral growth.
So again, going back to some questions about other different types of coral that we may be focusing on. So our modules are focused on the massive bouldering coral, which is, again, most of our reef building species in Hawaii are that shape. But we also wanted to look into branching corals. So we can't micro-fragment them and put them on our modules. It's not the same. So one of these species is very common on our reefs, Bacillopora demicornis, or lace coral. And so luckily, this coral has an interesting method of asexual reproduction. It produces planula, so coral larvae, or baby corals if you want. They kind of look like little seeds. And that's kind of what they are, is the coral will produce these planula. These planula will go and settle and transform into a coral, and that coral will grow. So with this species, Bacillopora demicornis, we're able to collect planula from individual colonies. And just like in our fast grow room where they're all genetically identical, it's the same thing with these planula since it's asexual reproduction. So we're able to isolate planula from one colony, and we're able to settle those planula on a concentrated surface, so say a tile. So we settle all that planula on one tile, and as they grow, they're all genetically identical. So their branches start to fuse when they get bigger. And so it's just another tool in the toolbox that we use to reaggregate corals so that we end up with a larger colony in a much shorter time than it would take in the wild for that coral to grow.
So here's another branching coral, Pocillopora grandis, or antler coral, that we find here on Hawaiian reefs. Now, if we were to grow, I'm going to go back. If we were to grow this species on one of our pyramids, it wouldn't serve the function that it does on a reef as a branching species. It doesn't have all the little hidey holes, and it doesn't provide the organisms that have evolved with it to reap the benefits that it has as a branching coral. So we thought, how do we use this species too? So one of our interns is working on coming up with a 3D module that we can cast in concrete that will grow micro-fragmented pieces of Pocillopora grandis that we can glue onto, there's our first prototype, onto our 1 foot tall concrete module that we can hopefully outplant back onto the reef that will serve its branching function. And also another event that we participated in with 7 high schools, it was called Coral Hackathon, where 7 different high schools had teams that came up with 3D module design prototypes. And the winner of the design is going to have their design 3D printed, and we will grow corals on that as well.
So as we were saying earlier, the state is really invested in this coral mitigation. So we're very excited to be expanding our coral nursery. We kicked ourselves out of our own office, and we transformed that room into another grow out room. So we have some tanks set up here. We have double the amount of tanks that we have in our other room. So that's really exciting. We will be able, with this new room, we will be able to produce 220 modules for our next project. And we also have a new building coming in August, hopefully. So we'll get our offices back. It's really exciting at the Coral Nursery. We're able to continue to expand and provide more for reefs. So we've had a lot of people work at the nursery. I just want to point out both past and present staff. We have many student volunteers, interns, as well as researchers who use and contribute to our facility. So we work closely with HIMB, so researchers can come down and use some of our research tanks and contribute to both their research as well as projects that we have at the Coral Nursery. And just one last thing, wrapping up. So kind of takeaway points from reef mitigation, especially in Hawaii, because our islands are so unique and our reefs are so vulnerable for reef loss. And we have high endemism of coral. Like we can't go to Australia and get replacement coral. So it's really, it's a really unique situation. So the state, we feel that it would be hard to get enough federal assistance to develop Hawaii-specific efforts towards climate mitigation and reef mitigation. So the state, particularly the Hawaii Coral Restoration Nursery, is really focused on this effort. So again, we are expanding our facility, and we're always creating new projects, and we're trying to come up with new methods to continue to come up with what's the best way to restore Hawaii's reefs. And so again, kind of the last sentence on here, if we don't do anything now, we might miss our window to help our reefs at all. So that's our last closing point. And thank you all so much. Mahalo. And if you have any questions, we'll take them now. Yes. Thank you.
Acclimation stage,
Mhm.
there's some fish in there.
Yes.
They're working fish.
Oh, yeah. They earn their keep.
So you moved outside, so it's starting to collect that algae and stuff. Is that what
Mm-hmm. Yes. So we have a lot of surgeonfish. I think there was like a yellow tang in that picture. So they are eating the algae that is maybe around the modules or on the platforms that we put the modules on. Yes, we do keep fish in our tanks. They're, as Honor said, they're working fish. So to try and eat algae or keep our maintenance down.
And since they are aquarium systems, we do get aquarium pests like Aptasia. So we have fish to control for Aptasia, which is an anemone that can sting corals. So we have fish that eat Aptasia too.
So then moving on back to your output location, I guess they're there.
Yes, so those fish are also present there. Yes.
I actually had I used to work on Bali, do the shark rescue.
Mm-hmm.
And there's these islands called the Gili Islands, and they got blasted, and from the fishermen.
Yeah, dynamite
And then all
fishing.
but the water's great, the diving, all the dive boats kind of smashed anything that was left. So they took the fragments. They started doing a bio-rock project.
Oh, mm-hmm.
And then the first time I brought sharks over, they're like, "You think of our bio-rock project?" I'm just like, and it was these French people. It's also kind of like, what kind of sucks, It had been overgrown with all the algae and seaweed. I remember diving it, and so it's these 3 circle islands, and it's pretty steep shelf. I remember there was no herbivore fish, so I was just free diving. I'm like, you know what? If I did, and I remember turning my back, and there's like big old torpedo. I'm just like, whatever. This was Hawaii. And then behind that, there was like a school of barracuda.
Mm-hmm.
And so I was like, they're just cleaning out all the herbivore fish. So then I remember telling them, you know what, I'll keep bringing sharks. Maybe it'll rebalance it. And so every 4 to 6 months, I get to go back with more sharks. And I started noticing all the herbivore fish came back. Just now it's pretty cool. It's flourishing, but it's a bio rock. They use
electricity.
Yeah.
Rebar, but it's fascinating.
Yeah. So herbivores are definitely important for coral reef ecosystems. They're the lawnmowers of the reef. Basically, they keep the weedy algae down. Yes, sorry, did you have a question?
Feeding them when you're rolling them up.
Mhm.
Is this include small animals for olives to catch?
Yes, so we feed a variety of foods. We have our live algal culture, so we have species of phytoplankton. We also have species of zooplankton that we can feed to our corals. We also have our, I like to call it coral smoothie. And we take frozen, basically frozen fish food. So usually a variety of shrimp. We have like mysid shrimp, spirulina, brine shrimp, and we blend that in a blender, hence the smoothie part. And so it's really fine pieces, and we use that for our coral food. So it's small enough that the polyps can grab that food and consume it. Sorry, did you have a read? Yeah.
Go ahead.
Almost done. Right.
I think you have quite a project going.
Thank you.
Thanks.
When you outplant, what's going to happen? How do you accommodate climate change?
Yeah, that's a tough question.
We continue to monitor our modules that we outplant, so we do check on them frequently.
Yeah, and then as
But-
far as like
Bleaching or anything.
we can't, there's no way we can prevent a storm coming through.
So where do you select? How do you outplant? What's your big plan?
Yeah, so we select our sites based off of where previous damage has occurred. So that is how we do our, for example, ship groundings is where we do a lot of our outplanting, like for example, the outside of the reef runway. And so we will go out and we will survey that site. We will look for appropriate places to place our modules. We will also look at the surrounding area to see what other corals are there. Just general observations to see, would this be a good spot for our modules? Is there, we don't want to say plant them in an area where we know they're just going to die. So, if there's a bunch of sediment runoff from an area, we're not going to want to outplant our modules there because just like natural corals, that, they probably wouldn't survive in a location like that. So we do survey our locations, and we do have careful planning to see what is the best location to outplant.
And also why we spend so much time and care getting the corals to that size where they can focus on growth and reproduction. So we do spend a lot of that time to get it 100% covered. So at that point, hopefully it can take what nature throws at it.
Do you guys have any human-related problems with your program? People going out and trying to harvest or anything that's going on out
Picking
there?
up the outplants.
Oh.
Try to pick out the outplants. They're
Yeah, they're on there.
You haven't had any human-related. You can have all kinds of related things. From
Yeah,
hurricanes and all.
no, we're not publicly like advertising. This is the GPS point of where we put our modules. So far we haven't had any issues human related. Yeah.
Second part of the question I was trying to ask was, your broad face, are the conditions that you're providing doing anything to help out the zones that the other two?
Mostly that would be like the light spectrum. So that's why we have those LED lights. And in those images, you can see that's kind of that blue cast. So we do have the ideal spectrum for the various clades of zooxanthellae. So when we are feeding the corals, that's kind of related to water quality. So we do track our water quality very carefully in our tanks, as well as we also track water quality of our harbor water, our filtered harbor water. We do track that so that we can have the optimal conditions for coral growth, including zooxanthellae.
And I'm sure that's just
Definitely no shortage.
You guys, for the lighting, do you mimic the sunrise sunset, or do you just leave them off for hours?
There's a full spectrum that it goes through.
Mhm.
Mhm.
Yeah, so our with our lights, they're really great. We're designed for the aquarium hobby, so that people on the mainland growing corals in their tanks would have, the optimal spectrum for their corals, for their home aquariums and public aquariums. So that's what they were actually designed for. So with our lights, we can change the spectrum. We can change the photo period. So the sunset, the sunrise, we have them gently ramp up in the morning to simulate sunrise. And again, at evening for sunset, we can also simulate different lunar phases.
What about the food that you guys culture for them? Are they on a fast growth cycle too?
We don't
By
really.
nature, I feel like
yeah.
they're fast growth cycle.
Yeah, we don't really need to try and fast grow them. They grow pretty fast on their own. Mhm.
For 24-hour photo period, help them to grow any faster instead of.
The coral?
Yep.
Well, so there's
The algae, that's used for phytoplankton to grow in 24-hour cycles.
yes. So like for example, our phytoplankton are grown on a close to 24-hour cycle, but zooxanthellae actually kind of max out. So there's actually a few interesting papers on the subject of different clades of zooxanthellae and what's the optimal photo period, which is again originally people looked into that for the aquarium hobby for growing their own corals. But yeah, they tend to max out, so they do need a rest period, just kind of like other like terrestrial plants where they respire at night.
If the city came to you guys and said, you have access to do work here at Hanauma Bay, would there be anything that you folks could do or would do?
We figured someone would ask. So yeah, we don't have any plans right now to outplant in Hanauma Bay, and we're not part of the city, and kind of we're focusing on coral mitigation. So we're not really trying to do policy or anything like that. But if we were to ever outplant in Hanauma Bay, it would definitely be, a coordination of various parties involved, and with a lot of careful planning.
Right now, we're focusing on mostly man-made damage. So if there was a real need in Hanauma Bay, then we would maybe consider having an outplanting site. But that would mean a lot of parties. Yeah.
I've heard some rumors, but what is currently being done to engineer corals for resisting climate change, if anything?
Do we have a thing about that?
We don't, but
Okay.
it's okay. Yeah, so for example, there's some labs at HIMB who are working on the genetics of different coral species. I think there was an article a little while ago that came out talking about super corals, and we're not really focused on we're not a research facility, so we're not looking into that. Those labs are the ones who are looking into genetics. But we also, we're looking at not just growing one type of coral or one species of coral. We don't want to, obviously then the people who look into super corals as well, we don't want to create a weedy species of coral, because then you're just going to have one type of coral. So we outplant a variety of species. Yeah. Does that answer your question?
Yeah.
Okay, great.
I think I missed it. How long have you been operating?
We're actually a relatively new facility. So we've been, well, the first part, we didn't have any corals yet. It was kind of building up the facility. We're just actually a little corner at Anuenue Fisheries Research Center. And so we kind of took over some old buildings in that area to build up our facility. So we're very young. Yes.
What, 5 years, 2 years?
Oh, yeah.
I think officially
I think it's
2015-ish.
Yeah, around there. Mm-hmm.
5 years, 2 years?
Yeah. So when we first, I
It's
feel like
June.
when you first start getting coral versus when you start building, we kind of think is kind of two different points.
Also, nobody else is doing this because corals around the world don't grow like Hawaiian corals. So our team had to develop whole new protocols and everything. So that took some trial and error too.
Yeah.
Sarah, I actually learned it in here. I think there was a coral bleaching cycle. Like 12.
A coral bleaching event is usually what it's like during summers.
I don't know if it was the El Nino, but it was like 15 years or 12 years. And then the guy said, but we're down to 6 years now. So that was where the big decline of corals, So if that's true, or whatever, if that's the trend, you guys trying to counter? I mean, how do you try and grow coral in warmer waters and stuff to try and
Yeah, I'm not super familiar with the concept of a bleaching cycle, so sorry about that. But we're not purposely growing corals in warmer conditions. Yeah, we're not trying to again, we're not trying to create like super corals or anything like that. We're focusing on the diversity of corals, the species of corals that we can outplant, and making sure that they're at a large enough size that even if they do experience partial mortality or part of it dies, that they're at a large enough size, again, that size refuge, that hopefully they can recover. Yeah, so again, we're not really a research facility, but we are working with some researchers.
And how do your corals compare to like, is it Waikiki aquarium like a bunch too, or is that more for aquarium trade?
That's more for their exhibits. They don't have a lot. They do have a representation of Hawaiian corals, but most of their corals that they have in the aquarium are probably most alike to South Pacific corals or Australian corals, and some Caribbean corals. They do have some representation from Hawaii, but it's for the aquarium. So they want the really beautiful, fast-growing species. Yeah, and you know, some of them are like purple and red, and most of the Hawaiian corals are like brown and yellow.
But
So
can you guys make a deal with those guys too, or?
that's our purpose with Maui Ocean Center.
Right.
Mhm.
Yeah. So so Maui Ocean Center is they have their rare coral arc just like we do, and they're also starting up a kind of similar land-based nursery as we do for handling Maui sites. Yeah. So we definitely share protocols and notes, and we just had some of their staff come and visit recently so that they could learn more about our facility, and you know, we send staff over to them so that we can learn about different techniques. So they're also starting up a similar nursery. Mhm.